Today, I am taking up the topic regulation of gene expression, and we will be studying about LAC Operon. In the previous classes, I have told you about the gene expression. Gene expression includes two steps. The first step is transcription. It means transcribing the information present on the template strand onto the mRNA strand. The mRNA strand undergoes some processing and then it enters the cytoplasm. In the cytoplasm, translation takes place. That is, from the information present on the mRNA strand, a protein or a polypeptide is synthesized. Then, how the gene expression is regulated. See, in all living systems, we find utmost economy. Energy is not wasted unnecessarily. Only the required substances are required at the required time. Then, for example, in human beings, there are 25 to 30,000 genes. If all the genes express themselves, or we can use another word, if all the genes are switched on or turned on, what happens? The cells will be filled with unwanted, unrequired substances. So, in living beings, it is avoided. It means the genes are switched on or switched off whenever it is required. Or, most of the time, most of the genes are switched off, and only the required genes are switched on for a specific purpose. It means there is some regulation in gene expression. Then the question arises, at what level this regulation takes place? Regulation can take place at many levels. It may occur at the level of transcription, or at the level of processing of mRNA in the nucleus, or during the transport of mRNA into the cytoplasm, or during the translation process of mRNA in the cytoplasm. So a regulation can occur at any level. But two scientists, namely Francois Jacob, a genetist, and Jacques Monod, a biochemist, they found out in the year 1961 that the regulation is at the transcription level itself. That means during copying the information from a template strand onto the mRNA strand. So, at the transcription level itself, genes are, or gene function is regulated. Their experiment is known as LAC Operon. I will come to that point later on. So, they made the discovery that genes are regulated at the transcription level itself in all the prokaryotes. I think same thing happens in eukaryotes also. For their work in 1961, they have received the Nobel Prize in Medicine in 1965. Here, LAC means lactose sugar. Lactose sugar is a disaccharide. It consists of one glucose molecule and one galactose molecule. Their experiment was on the bacterium E. coli. E. coli usually feeds upon glucose. When it feeds upon glucose, these three enzymes are not required. It directly utilizes glucose in its metabolism. When these three enzymes are not required, the structural genes are switched off. When the E. coli is given lactose sugar, that means the lactose sugar has to be split into glucose and galactose. For that purpose, it synthesizes all the three enzymes. How it happens, I will tell you just after some time. Then, the model proposed by Jacob and Monod is known as LAC Operon model. What is an Operon? You should know. Operon is a group of closely linked structural genes and their regulating elements or regulating genes. So there are 2 points, structural genes and also regulating genes. Then what are the structural genes? Structural genes participate in the transcription and decide the amino acid sequence in proteins. That means they participate in transcription of mRNA. Transcription of mRNA. The regulatory genes. The regulatory genes control the activity of the structural genes. So, an operon consists of structural genes and also their control genes. In the case of LAC operon, there are 3 structural genes and 3 regulatory genes. The structural genes are always remain together. They occur continuously. Just ahead of the structural genes, we find the regulatory genes. The regulatory genes are 3 types here. I gene, P gene and O gene. I gene means inducer gene, inducer. This gene is responsible for the production of a suppressor substance. It is responsible for the production of a suppressor or inhibitor substance. P gene means promoter gene. It is the site at which RNA polymerase enzyme is attached for transcription. So, at the P site, RNA polymerase enzyme gets attached for transcription. Then O gene, that means operator gene. The repressor substance produced by the I gene gets attached to the operator gene. Whenever transcription of these genes is not required, that means when the repressor substance gets attached to the O gene, transcription comes to an end or transcription fails to take place. Then, structural genes. These are 3, Z gene, Y gene and A gene. They have different functions. They together produce one continuous mRNA strand on transcription. And this strand is known as polycistronic mRNA. So here 3 genes are continuously producing one mRNA strand, and that mRNA strand is known as polycistronic mRNA. Then the polycistronic mRNA enters the cytoplasm, or of course in the bacteria there is no difference between this one. It divides into 3 parts or 3 segments, and the mRNA produced by the Z gene is responsible for the production of the enzyme beta galactosidase. So the gene Z produces the enzyme called beta galactosidase, and its function is to cleave lactose into glucose and galactose. That is the function. Then Y gene. The mRNA produced by the Y gene causes the production of another enzyme known as the permease enzyme. Permease means this enzyme allows the bacterial cell the lactose sugar into the bacterial cell. So to enter the lactose into the bacterial cell, this enzyme is required. Without this enzyme, lactose sugar fails to enter the bacteria. Then, A gene. It is responsible for the synthesis of the enzyme transacetylase. And this enzyme transports an acetyl group from acetyl-CoA to beta-galactosidase. Then, beta-galactosidase has another function also. It converts lactose into L-lactose. Is very important. L-lactose is very important. It binds to the repressor and detaches it from the operator gene. I will tell you here afterwards. Always some L-lactose or lactose is present in the bacterial cell. Then, coming to the LAC Operon, I have told you, when only glucose is given as a food medium, these three genes are blocked. They do not produce any mRNA. But when the E. coli is raised upon lactose sugar, these three genes are activated. How it happens, now we will see. This is the first figure. This is the DNA segment. These three are the regulatory genes: inducer gene, promoter gene, operator gene. These three are the structural genes. Then, in the absence of the inducer, here inducer means lactose. So lactose sugar, when we introduce, this LAC Operon comes alive. So here, inducer means lactose. In the absence of lactose, what happens? The I gene transcribes one mRNA. This mRNA produces a repressor protein. This repressor protein goes and attaches to the operator gene. When the operator is blocked by the repressor protein, transcription cannot occur. Transcription fails to occur here because RNA polymerase enzyme fails to attach to promoter. For transcription of this information present on the three genes, mRNA polymerase must get attached to the P gene. It happens only when the operator gene is not blocked by the repressor protein. So, only in the absence of the repressor protein, mRNA polymerase attaches to the P gene and transcribes them. Now here, the operator gene is blocked by the repressor. So, transcription cannot occur. But once we introduce lactose into the food medium, what happens is, the L-lactose, I have told you, always some L-lactose is present in the cytoplasm of bacteria. It combines with the repressor protein. When L-lactose combines with the repressor protein, structural changes occur in the repressor protein. And the repressor protein is removed from the site, from the operator. So I will tell again. L-lactose combines with the repressor protein and forms a complex known as repressor allo-lactose complex, and this allo-lactose brings about structural changes in the repressor. As a result, the repressor leaves the operator site, and this repressor allo-lactose complex goes into the cytoplasm. Now, operator gene is free. It is free from repressor protein. Now, RNA polymerase comes and attaches to the gene P or promoter gene, and it transcribes the three genes together. So three genes means Z, Y, and A. As a result, a polycistronic mRNA is formed. A continuous mRNA for all the three genes is produced, and it is known as polycistronic mRNA. Then, this is transcription. Then translation. the polycystic mRNA breaks down into 3 segments, and these 3 segments will produce beta-galactosidase, permease, and transacetylase, as already I have explained. I have also explained their functions. Again, I will tell you. Beta-galactosidase, its function is cleaving or splitting lactose into glucose and galactose. It also produces allolactose. Permease enzyme permits the lactose sugar into the bacterial cell. Transacetylase, it helps in transferring CoA enzyme to beta-galactosides. So in this way, transcription takes place and translation also takes place. This is the explanation given by Jacob and Monod about LAC operon. It's very simple. Then, gene regulation occurs in 2 ways again. One method is called the negative method, another one is positive method. LAC operon is a negative regulation. In all the prokaryotes, regulation is usually negative. In all the eukaryotes, it is positive. Here, negative regulation means operator gene is blocked by the repressor protein. Positive regulation means a transcriptional factor is required to be bound to the promoter. So, in the case of positive transcription regulation, one transcriptional factor has to come and attach to P position or P gene. Then only transcription occurs. So in this way, gene regulation is either negative or positive, and in the case of LAC operon, it is negative regulation. Then, when all the lactose present in the food medium is exhausted or used up by the E. coli, then what happens? In the absence of lactose in the food, again the gene I is activated. It transcribes mRNA, repressor protein is formed, and it goes and attaches to the operator gene, and thereby transcription comes to an end. So in this way, LAC operon operates in bacteria, and the example used by Jacob and Monod is E. coli. So I have written all these 3 books. If you are interested, you can buy them from Amazon. They are only kind literature books. Thank you.